Hello, methylobacterium.
Who are you?
I am a student in OEB 100. I've been waiting for you. You are a bacterium, one from a population of several million that we have been studying this semester. More specifically, we've used you to learn about microevolution, how bacteria, when exposed to an environmental stress, will grow more slowly or die for the most part, but that some survive.
Why am I here?
Your life is the result of microevolution. You are here because despite exposure to a harmful stimuli, one of your ancestors developed a mutation that conferred some level of protection to every bacterium born from this ancestor. This mutation is an adaptation and is the result of rapid division and imperfect DNA replication. It is what has allowed you to survive and what we are looking to study. We want to find out more about what this mutation changed, how it affected your ability to survive in different types of harsh environments.
This past year, our lab has been working to examine the growth of Methylobacterium extorquens on formaldehyde. We know that formaldehyde is an intermediate in the metabolism of single carbon compounds in Methylobacterium. Thus, though it is a toxin, formaldehyde is also a form of food for the Methylobacterium. We tested the bacterium's ability to grow in formaldehyde by gradually increasing the concentration of formaldehyde each time we grew it up. Eventually, we had Methylobacterium that could successfully grow at high concentrations of formaldehyde. These plates show that we evolved 4 replicate populations to grow on formaldehyde, but could only get populations 2, 3, and 4 to form colonies on solid media. Hence, our work this semester only focuses on isolates from populations 2, 3, and 4.
I know Kung Fu.
Show me.
When organisms evolve new traits, sometimes they can lose other abilities in the process because there are trade-offs between developing new traits and maintaining others. A change in one gene may be beneficial in some circumstances but have a negative effect in other environments. On the other hand, a new mutation can confer multiple abilities, and the development of formaldehyde resistance may result in a general ability to withstand stress.
Good. Adaptation, improvisation. But your weakness is not your technique.
We wanted to see if there were any trade-offs occurring in the evolution of Methylobacterium extorquens to eat formaldehyde. We grew up the isolates and the ancestor in several different environments and then tested their abilities to grow under stressful conditions. If evolution on formaldehyde resulted in trade-offs, we would expect to see differences between isolates and the ancestor under these conditions.
Again.
We used a robotic apparatus to measure the growth rates of our Methylobacterium. The robot measured the number of individual cells to determine how fast they are able to divide under different conditions. We found that exposing the Methylobacterium to high concentrations of formaldehyde decreased the growth rate.
In my experiments, I wanted to examine the growth of formaldehyde under different ionic stresses. Testing growth under pH stress allowed me to see how the bacteria would grow in different concentrations of hydrogen ions. Salt stresses allowed me to see how the bacteria would grow in different concentrations of a variety of salt ions. After growing the Methylobacterium in either formaldehyde or methanol, and then testing their growth at different pHs, we observed that the bacteria grown in formaldehyde grew better under pH stress. This suggests that Methylobacterium grown in formaldehyde have some sort of increased resistance. The salt experiments so far have involved plating different isolates to see how well the bacteria grown in methanol versus those grown in formaldehyde grow at different salt concentrations.
Since developing the ability to eat high concentrations of formaldehyde must have involved developing some resistance to the normally toxic substance, we wondered if evolution gave Methylobacterium resistance to similar toxic compounds. Glutaraldehyde is another very toxic substance. We grew the isolates and ancestor in 2 different environments, either eating formaldehyde or methanol. Then we grew the bacteria with different amounts of glutaraldehyde and looked for different amounts of growth. The ancestor wasn't able to grow in the presence of glutaraldehyde, whereas the isolates could grow at varying concentrations depending on environment. This shows that evolution on formaldehyde improved the ability of methylobacterium to deal with glutaraldehyde stress. It also shows that environment can make a difference in stress response.
When the cells were first grown in formaldehyde, and these mutations became fixed if they were beneficial in the presence of formaldehyde. This is evidence of adaptation and evolution. Second, selection for the ability to grow on formaldehyde resulted in many different adaptations in the isolated populations. Every isolate is like a snowflake, unique, even though they all evolved under the same conditions. This shows that there are many different evolutionary trajectories accessible to organisms, and that there is not a predetermined course of adaptation. Furthermore, we explored the principle of trade-offs and found that an adaptation can be beneficial under some circumstances but have a negative effect on fitness in other environments. Next, we will investigate the adaptations on a genomic level by sequencing the genomes of the isolates and looking for differences between the isolates and the ancestor. Then we can insert genes from the evolved strains into the ancestor and determine the role of each mutation.
I know you're out there. I can feel you now. I know that you're afraid. You're afraid of us.